Very happy to introduce today's TEOS speaker. I've never actually hosted a remote seminar here for TEOS. This is my first. Professor Delphine Farmer from Colorado State. I can't help but smile when I talk about Delphine. She's just such a wonderful colleague. She's also a co-principal investigator on a big project that we're spearheading here at Stony Brook called GOTHAAM, which was supposed to go this past summer, but it will go in the future. So Delphine has been an integral part in the planning of that project. She has a lot of field experience. She was a participant in WECAN, which was a biomass burning fire experiment using the Hercules C-130 aircraft. She's got a lot of flight time, a lot of observational experience. She really focuses on aerosol chemistry and composition, the organic component of aerosol. And she's most recently been working on not just combustion and biomass burning, but also indoor air quality as well. But today I think she's going to be focusing on wildfires. Delphine has a big group. She's prolific. And I think actually Daniel, one of your former graduate students, is now a postdoc with Delphine. So we've been interacting with Delphine's group for some time. If anyone has any questions, Delphine said you can shout it out if you like, but maybe if you want to put it in the chat, we can make a record of it. And in the interest of just staying on schedule, maybe we can have a discussion towards the end. If you have something pressing, of course, feel free. Otherwise, please mute your microphone and let's hand it off to Delphine. I don't know how we're supposed to clap and welcome you online, Delphine, but welcome.
I know that's what's going on. I hear you. Thank you.
We're all stoked, so take it away.
Great. Well, thank you so much. I really appreciate the invitation here. I'm delighted to virtually come to Stony Brook. I've been on campus once before, so it's really nice to at least be able to come and share some of the science we're doing. So what I'm going to talk to you about today is some of the work and how I've just been thinking a little bit about wildfire smoke over the last few years. And obviously this is an emerging problem and an emerging question. And I've really come to the conclusion that the wildfire smoke is a bit of a villain in the atmosphere. It brings a lot of air toxics, a lot of challenges and it transports them across the globe. And so I'm going to tell you a few stories a little bit about how I think about wildfire smoke and how it moves through air, leaves, and homes. And we're going to do this a little bit by thinking about three different stories, and these are basically three different villains in the atmosphere. So as I say, we'll think about air first, then we'll think about plants, and then we'll think about what happens when the smoke gets indoors. But the piece of background I want to start with is this challenge that wildfires and the smoke associated with them are increasingly prevalent. Dan Jaffe did this lovely analysis of data. I really like the figures in this paper. And what he was able to show very clearly and elegantly is that if you look at particulate matter in the US and you look at trends from the 1980s to 2016, you see the particle sulfate on the bottom has generally decreased. All the colors in greens and blues show decreases in particulate sulfate. And that's because the Clean Air Act is an amazing piece of legislation and actually works. The challenge is when you look at particulate carbon, and that's the map on the top side, what you see is that in the Western United States, particulate carbon has been increasing dramatically, and that is really due to wildfires. And this influences both outdoor and indoor air quality, and we'll get into both of those in this talk. And then the reasons for this are really that wildfires are influencing the air quality, and that's driven by climate change, obviously, by human behavior and land use and forest management practices. Okay, so the reason I'm interested in wildfire smoke and atmospheric chemistry in general is that changes in the chemical properties of the atmosphere really affect its optical properties and the quality of the air that we breathe. And these two pictures, I think, highlight that very well. They're both pictures from Colorado. And what you can see on the left is the city of Denver. And this is in the summer. There's this brown cloud that's known to sit over Denver. And you can see that trace gases are absorbing light, and the particles are in there as well. And so you see absorption and scattering. So you see climate effects, but you're also seeing air quality effects. You're visually seeing the NO2 that's a known air toxin. On the right, you see some pictures of wildfire smoke plumes. And what you should be able to notice is that those plumes, which have a huge number of particles, can then pull water out of the atmosphere and grow into water droplets and form some very beautiful white clouds. And this is the pyrocumulus clouds are just a very dramatic example of how aerosols can change the indirect effect and can change the optical properties of the atmosphere. So this might be the background, but at the end of the day, what we really need to think about is the climate, ecosystem, and human health effects of all of these different trace gases and particles that we're putting into the atmosphere. And that effect depends on the concentration, right? And the concentration is dependent on the sources and sinks, the balance of how much is put into the atmosphere and how much is removed. So we spend a lot of time thinking about sources in the atmosphere, and what I will argue today is we don't spend enough time thinking about their sinks. But that balance is really important. So there are some ways we can think about that, and this is one way. So we can think about primary emissions, so that's direct sources of, say, particles or reactive trace gases, and there are natural sources and there are anthropogenic sources. And then there is secondary chemistry. So there's chemistry in the atmosphere that produces more particles, changes those gases, and then changes those concentrations. So those are various sources. Then as air is transported downwind, and I think you all had a pretty dramatic example of that with wildfire smoke from Canada moving over to New York this summer. During that transport process, there's a lot of chemistry. But towards the end, to remove those components from the atmosphere, we have two major pathways. There's wet deposition, so precipitation, rainfall, snowfall, pulling out trace gases and particles. And then there's dry deposition. And so that's when particles or gases effectively stick to surfaces. So what my group really does is we use field measurements to try and understand and constrain the magnitude and the drivers of all of those sources and sinks of reactive trace gases and particles. So I'm really interested in basically these arrows, the processes that control them. And ultimately what we're trying to do is constrain the impact of these different sources and sinks on climate, ecosystem, and human health. And that ideally will allow us to identify strategies to reduce those impacts. And I think this is representative of the entire field of atmospheric chemistry. So the question we're going to address today is, how do gases and particles from wildfire smoke interact with different surfaces, with forests, and with buildings? So we're going to focus on the wildfire smoke source, and then we're going to think about different sinks for each of these, for different components of wildfire smoke and particles in general. So the way I got into this, and John mentioned this in the introduction, was that I was involved in the WECAN experiment. So Sonia Creighton-Weiss at CSU and I were actually funded to do aerosol composition measurements. We were initially going to do this on a small plane, but as you may have heard from GOTHAAM, plans with research aircraft tend to change a lot. And we ended up hopping onto NSF C-130 aircraft in 2018. And we flew out of Boise, Idaho, and spent a lot of time trying to go into wildfire plumes. And so you can see all the different flight tracks and all the different fire plumes that we directly flew into on the map here. And this was a really great experience. We learned a huge amount, and I have to call out my research scientist, Lauren Garofalo, who really took the lead with trying to get excellent measurements and high quality measurements from an extremely complicated instrument. And I have many stories to tell about WECAN, both science and otherwise. but I'm just going to leave this with one figure, and that is that when we take all of these different wildfires and we look at the first pass, the closest pass to the wildfire plume, and so we look at those initial emissions, what we find is that submicron aerosol and wildfire smoke is overwhelmingly organic, well over 90%. There are trace amounts, and they're extremely important, of different types of inorganic components, but most of what we're looking at is organic aerosol, and then there's a huge amount of organic gases that come along with this aerosol. So that's what wildfire smoke is. And of course, we were flying up high, but this started getting me interested in thinking about what happens down on the ground. So wildfire smoke includes a complex mixture of gases and particles. It's mostly organic, and it does all sorts of strange chemistry. So the first story we're going to talk about today is the Grinch who stole particles. And I'm just going to take a moment because my computer is threatening to restart. So hopefully I can stop that from happening. It's a little scary. All right, we're just going to go with it. All right, so the Grinch who stole particles. So the first thing, which we're gonna see, there we go, is that the Earth's surface, as I say, it steals aerosol particles from the atmosphere. That's deposition processes. And these loss rates are really important because the loss rates of this aerosol actually define the atmospheric lifetime of the particles. And I think that's something that's not necessarily appreciated as much as I wish it was. Our atmospheric lifetime, so how far can a particle transport, how long it lasts to do something, is entirely determined by its loss rate. So that's the Grinch. And this is important because particles are the strongest contributors to uncertainty in radiative forcing estimates. And you can pick your favorite IPCC report, but this is a really nice figure in AR5, and what you see is that if the effective radiative forcing on the x-axis, obviously anything to the right, so anything positive has a warming effect, and that's greenhouse gases drive that. The uncertainties on greenhouse gases are actually very small and quite well understood. And then you look at this huge range of uncertainty on the negative side, and that's the cooling effect that's driven by aerosols. And so when we look at how humans are changing climate, and we think about this in terms of radiative forcing, then what you see is this large uncertainty that is driven by aerosols. And of course, this is challenging because right now people are very interested in understanding regional impacts of climate. And that is driven by has strong effects of short-lived climate forcers. So understanding how aerosols work and what it is they do and how long they last is obviously very relevant. Okay, so then we can look at where all of these uncertainties come from. And this is this lovely study from Ken Carzaw's group in Leeds. And what his group did is look at all of the different things that contribute to that aerosol radiative forcing effect, and how well do we know each of those components. So if we look at this figure, it's basically a metric of uncertainty in the ability of particles to form clouds, and in that cloud condensation nuclei. So those clouds those particles that form nice puffy white clouds. All right, and when you rank all of these, what this group found was that the single largest uncertainty was actually in dry deposition. And so that's of accumulation mode particles. So these are particles that are between about 100 and 1,000 nanometers, so submicron. So conveniently happens to be right in the size range of all of the instruments that my group has. And what this says is that dry deposition is the single strongest individual source of uncertainty in cloud condensation nuclei. So this looks to me like this was a thing that we should start really thinking about. So I worked with a couple of students. The first one is Erin Bodecker, and Erin spent a lot of time helping me think about processes that control particle dry deposition. And so the first one is really just gravity. So gravitational settling pulls particles out of the atmosphere, very simply. And obviously the effect of this, effectively the dry deposition velocity for the particles depends on the particle size, and it's very simple. And we're not going to argue with gravity. We decided that the uncertainty on that was small, and we weren't going to try and spend too much time reconsidering that. Brownian diffusion is really interesting. This is a process by which particles just move through and diffuse through the air. And if they happen to use this process and then hit a surface, then that can lead to deposition. And Brownian diffusion leading to dry deposition is generally thought to be most important for very small particles. So particles less than 100 nanometers in diameter. The next process is impaction. And what impaction is when a particle moves against the currents of air and hits the surface. And so impaction generally thought to be more important for larger particles. And then the last process is interception. And what interception is when particles are pulled with air currents and then they pass along different surfaces like a leaf surface and then stick to that surface. So it's the balance of these 4 different processes that give us this very distinct theoretical curve on what particle deposition velocity would be. So what this tells us is that the smallest particles should get pulled out quite rapidly, right? The Grinch is going to steal those. And then the larger particles are also going to be pulled out quite rapidly. What the Grinch kind of ignores are these mid-sized particles in the 100 nanometer to 1,000 nanometer range. But remember, these particles are actually really important. They contribute to cloud condensation nuclei, they contribute to air quality issues, and they're very poorly understood. So what my group did was spend some time thinking about size-resolved particle flux measurements. And so the first site we went to was Manitou Forest. This is a pine forest in Colorado. And there are lots of beautiful birds for Ernie. What the site is represents much of the United States, but certainly much of the Western United States where you have conifer forests. So we spent a lot of time, several seasons out there. We also spent time at the Southern Great Plains site in Oklahoma. This is basically a grassland, an alfalfa field site. And so we just spent a few weeks there in 2016. And this work was done, a lot of the measurements and this initial analysis was done by Ethan Emerson, who was a PhD student of mine. So when Ethan took all of this beautiful data and, you quality assure and quality control, and so you lose large amounts of it, but you take what you have and you average it out. And we wanted to first compare it to a model. This is the disappointment that we had. So you have particle diameter on the x-axis, you have deposition velocity on the y-axis. We took the two parameterizations really that people widely use in pretty much all models for particle deposition. Those are those lines. So we have one for evergreen needle leaf forests and one for the grasslands. And then we take our data and we put our data on. The only thing that looked about right was that we had faster deposition over the pine forest. And that should make sense because there's so much more surface area with all of those needles in the forest. So we should be seeing faster deposition, right? More impaction, more interception, there's just more surface area. So that made sense, but nothing else looked right. So we spent about a year going back into the data and checking that we had done the analysis right, that the error bars were appropriately calculated, that we met all the turbulence conditions, and we were pretty convinced that that our data were correct. So then we started revisiting the models. And we also did what every graduate student should do in this situation, which is take a look at the literature. And once we put our data in the context of the literature, we suddenly started to feel a lot better. So this is the figure that Ethan put together over time, and then Aaron Bodeker added some data points. And so again, we have our particle diameter on the x-axis, deposition velocity on the y-axis. And what you see is the model in the dashed line, and all these different forest observations in all the different markers consistently do not agree. And our data are those red triangle, red diamonds in the middle. They just sit in the middle of all of the literature. So we're consistent with previous work. We found we weren't the first people to notice that there was a problem with the measurements in the models. There were some other papers that had done that. But we wanted to then think if we could use our particularly large data set to improve how we parameterized dry deposition. So Ethan Emerson spent a bit of time thinking about this, and what we came to the conclusion of is that current models are overestimating Brownian diffusion. At first, the role of Brownian diffusion in terms of enhancing dry deposition. So not to say it doesn't happen, and we're not about to change Fick's law, but we don't think that that's as important in terms of a loss process as the current models describe. And then the other thing we noticed is that interception, again, this process of particles moving with air currents around surfaces could actually be more important than had previously been estimated. And I'm not going to go into all of the detail of how we came to these conclusions, but it looks like once we pull these two parameters out and we just change their relative contributions, we can then go ahead and start thinking about a new parameterization. And so what we have on the left is a parameterization in the shaded regions, as opposed to the all of the observations from the literature in black and from our data in red. And so on the left, we have a needle leaf forest. And what you can see is the original parameterizations failed to capture what it is that the observation showed. And once we, again, dialed down Brownian diffusion, increased interception, we find that we can match our model, our measurements much better. And we also decrease the uncertainty. Grasslands, there were fewer data sets out there, but it's a similar story. Marine systems are actually very interesting because models assume no interception, because oceans are assumed to be a flat surface. And so the changes over the oceans have a slightly different structure to them, because all we're doing is reducing that role of Brownian diffusion. So once we have this new parameterization, we were able to call some of our friends here at CSU who are in the Atmospheric Sciences department, and they could start to think about what this did in terms of modeled impacts on aerosol concentrations. And so there are two pieces to notice here. One is that small particles, we think, actually deposit slower than most of the models currently estimate. And then we think that larger particles, so above about a micron, happen to deposit much faster. So we've raised those deposition rates. So we tuned down the Grinch in small particles, and we made them a little more impactful on larger particles. Okay, so if we take those small particles, this is fine. They exit the atmosphere more slowly. So there are more of them. So we see increases that are on the order of anywhere between about 10 and 100%. Increases in small particles, dramatic effects over the ocean, which I don't like reading too much into because there are so few observations of particle deposition over marine systems that have size resolution that this is very hard to actually validate. But certainly over the land, we still see this effect quite strongly. All right, then we can think about particles in the 5 to 10 micron range. So larger particles, they exit the atmosphere much faster. And so there are fewer of them. So over land, we see far fewer large particles in the models than we initially predicted. And again, marine systems are different because they assume no interception. And so I leave that as a large question mark because I'm not sure that that's actually true. But we can think about what's happening over the land a little bit more. And so we can actually think about the indirect effect. So that's that if we're changing the number of particles that are potentially going to form droplets in clouds, then we're going to change that indirect effect. So Jeff Pierce's group at CSU was actually able to calculate this. And so how much we were really changing that aerosol indirect effect merely by changing the parameterization for particle deposition. And if we just focus in on the land, because that's the parameterization I trust more, we still see this negative effect. And this is aerosol effect. This is not radiative forcing. So this isn't the change since 1750. This is the change at a given point in time now. But we do see a very substantial effect. So what this tells you is twofold. One, it tells us that particles, again, those particle loss rates really matter. They change the concentration enough to have a radiative effect. The second thing it tells us is that this is something that perhaps we should be getting right in models. So we can think about this from different perspectives. There are aerosol indirect effects, there are direct effects, and those are a little more complicated because different sizes of particles play different roles in different locations. And then we can also think about health effects and PM 2.5. And so if I draw your attention to the model output on the far right, you see that PM 2.5 over many places actually increases over many land areas, although certain distinct regions, mostly heavily forested regions, we see an enhancement in deposition. And so we see lower PM 2.5. So what hopefully you get out of this is that those deposition processes impact climate and health predictions, but they are often incorrectly described. And new observations allow us to do better and with less uncertainty. So truly, I will say the land surface is like the Grinch. They pull out all of those particles, but actually at the end of the day, there's some good things that it's doing. So what are we doing now? I don't have many results. If you come to AGU in a couple of weeks, my postdoc Joshi will be able to tell you about some of the interesting results we have. But what we are doing is now realizing that we need to make many more measurements over many more land surfaces to constrain these processes. So we are funded by DOE for what we call the Falcon Project to look at fluxes of aerosols in a continuous observing network. And so what Falcon has us using is it's lower cost sensors. They are not cheap, but they are not, full aerosol spectrometers. And we have set up flux measurements we're currently running over 3 different surfaces. We have a flux system out at Scripps Pier in La Jolla getting a coastal marine measurement, which obviously we need to we've got some ideas about how to subtract sea spray emission from that deposition process, but the data is looking promising. We have a system back out at the Severn Great Plains site in Oklahoma, and we are out in Oak Ridge in Tennessee at a NOAA site, again, looking at fluxes, and then we're looking to deploy out at Barrow and down in Alabama as sites come online. So this is the plan. So hopefully we'll have a little bit more observational constraints on these deposition processes soon. So that's the particle loss story. The next thing is this question about what happens to plants. So we deposit particles, and maybe we think about these smoke particles. We deposit them onto forest surfaces. What happens to them? And what happens to those leaves? And it turns out to be kind of interesting. So this site out at Manitou Experimental Forest, where we did particle deposition measurement, we've also been going back out there repeatedly to think about carbon measurements and VOCs and various trace gases. And so Dylan Millay from the University of Minnesota and my group got together in what we call the Fluxes of Carbon Study, or flux, in 2020 and 2021. And we aim to study the VOC exchange over a Colorado pine forest. So Dylan Millay brought a proton transfer reaction mass spec so that we could look at all of the slightly more reduced gases. My group has a chemical ionization mass spec, which we use to look at a lot of the more oxidized gases. And we were able to put these instruments together and then also pull some offline measurements and start to think about how leaves emit and take up different types of organic carbon. And so the first part I want to talk about, or what I'm going to talk about here, is some work done by MJ Riches, who did her PhD in my group, and she is now an NSF postdoc here at CSU. And what she was thinking about were these leaf level measurements of plant physiology and VOC emissions. And so the way we can think about this is if you take a ponderosa pine tree, so an example, and you look at just some of those individual needles, there's a lot of interesting things going on. On the bottom of those needles, there are actually stomates. So these are basically holes in the leaves that the plant can open up or close. And when they open up, they can pull in CO2, they can release water, and then they happen to exchange volatile organic compounds at the same time. And so that's one process by which ponderosa pines release that very characteristic pine scent, which happens to be driven by a bunch of terpenoid volatile organic compounds. I'll show you those structures later. The other thing that's important about ponderosa pines, though, is they have these resin ducts inside the leaves, and those hold on to all sorts of volatiles. And then those volatiles can diffuse out the leaf and then be released. So MJ has this LICOR. It's a standard plant photosynthesis measurement where we can exchange and look at CO2 and water. We modified this so that we could then also pull some gas phase samples out at the same time. Okay, so what we're going to think about are these two processes. Plants emit VOCs through both the stomates, incidentally also referred to as stomata, depending on where you went to school. But we're going to move through the stomates, and then there's also, as I say, diffusion from the leaves. There's also sap, bark, and flowers. We're going to ignore those for right now. Okay, so what kind of VOCs are we looking at? Turns out there's a lot of small reduced molecules, and then there's some oxygenated molecules. So we were able to make measurements of quite an array of these. And what we were really doing, let me go back here for a moment, was really thinking about just how temperature, humidity, light, CO2, how environmental parameters would influence that exchange. But as Colorado is in the summer, and as I showed you on the first slide, we have an increasing number of wildfires in the region. And so poor MJ Riches was taking measurements when we got a massive set of wildfire events through Colorado. And so you can see this picture in the background, this picture from our site. The sky was covered in brown and orange, and the PM 2.5 levels were enormous. So my students were wearing masks because it was fall 2020 during the pandemic, but we also were concerned about what this would do to the plants, and it turned out to be really interesting. So what happens is when you have a lot of smoke, the plants close their stomates and they basically get stuck. So even when the smoke cleared, those stomates wouldn't open back up. The plants didn't, under any conditions, any standard conditions, the plants wouldn't start exchanging CO2 or water again. So they were stuck. So this is the first period. Stomates were closed, plant wasn't productive at all. We then started to increase the temperature to induce stomatal opening. I will say there were a lot of cell phone calls between MJ in the field and me on a Saturday morning trying to figure out how we could get the stomates to start doing what they were supposed to do so that she could continue on with her experiments. And once MJ had really ramped up the temperature, so sitting at about 40 degrees centigrade, then we could start to slowly open up the stomates. Then by cutting down the humidity dramatically, the stomates suddenly burst open. And what happened when they burst open, let me just pull this here. Okay, when the stomata burst open, after we decreased the temperature and relative humidity, then we could start to see, again, photosynthesis occurring and gas exchange. So initially, in this process, the smoke gases, we think it's the gases, it could have been the particles, but we have some evidence that it's the gases inside the plant stomata cause those stomata to stick. They close and we reduce plant productivity. Then when we open the stomates, we see, what we end up seeing is a release of a huge amount of VOCs that have been trapped inside. And so we can walk through a few of these examples. So what I'm going to do is show you this figure on the top left, and you just see a function of time and minutes, and you're going to see a chemical tracer. So this first example is nitrophenol. And what was happening is that while MJ was trying to figure out how to open the stomates, nitrophenol, which is a smoke VOC, was in the air, and it would slowly get taken up by the surfaces of the leaves. The stomates weren't open, but the leaf surface could take up those gases. Then once the stomates opened, that sudden burst at 0 minutes in the middle of this figure, we saw this huge belch of nitrophenol and a bunch of other VOCs. And so what we think we were seeing is that smoke VOCs had partitioned to the stomatal cavity during the smoke event before those stomates locked up and closed. We see uptake of nitrophenolics and other smoke VOCs on the leaf surface. But then as soon as we could open up the stomates, we would see this long sustained emission. And it looks like that reservoir was going to deplete in about an hour. So there were a fair amount of nitrophenolics that had stuck to the surface inside the leaf and then were coming out. All right. Next type of VOC, there was a set of VOCs. We've got acetic acid in black on this next figure and methyl salicylate in red. And what we saw was a set of VOCs that did not, there was some slight pre-burst uptake of some of the organic acids, so things like acetic acid and maleic acid, but generally near zero. And then as soon as we had that stomatal burst, these VOCs would rapidly release, and then they would rapidly go back down to zero. And methyl salicylate is a stress marker for plants, so we think that probably the pines did not like being exposed to smoke, and so they created this stress marker, and that accumulated inside the stomatal cavity, and then when we finally were able to open that cavity, all of these smoke VOCs came out. Okay. Then we saw what we thought is a surge emission. So this is a set of compounds. Methyl isocyanate is one example. This is a smoke VOC again. And so this was set of smoke VOCs that were trapped inside the stomatal pore. And once we, again, open that stomatal, open the stomates, they would come out in a burst, but then we would have this constant emission source. And so we think that either these are isocyanates that are being formed on the surface inside the stomatal pore, or they were formed inside the leaf and are just being slowly released out over time. So this is not a compound that one normally sees in biogenic natural VOC exchange, but it was something that we saw pretty constantly after large wildfire smoke events. And methyl isocyanate is a pretty nasty toxin that is known in terms of its role in the Bhopal disaster in India and other industrial accidents. So this is a smoke marker that is not healthy for the plants. The last type of sort of VOC that we saw was this post-burst uptake. So we would see, we saw hydrogen cyanide, which is a smoke marker, and we would see the hydrogen cyanide and a few other organic acids be taken up by, again, the surfaces of the leaf. And then when we open those stomates, we would see a tiny emission, but then we would just see a huge amount of uptake. So this tells us that some of these compounds are ones that once they get into the plant, they are not going to be released. So the plant just continues to take up dominantly through the stomates some of these smoke VOCs. Okay, so one last note is that we saw some interesting effects on temperature responses. And so we know that ponderosa pines, much like many other plants, if you increase the temperature on a normal day, plants will then just release more and more biogenics. And we would see that with monoterpenes. So in this figure, you see air temperature on the x-axis. You see on the left-hand side and in the black line, solid line, you see a total monoterpene emission. And then you see that photosynthesis decreases with temperature. This is actually normal behavior for ponderosa pines. So this is what we normally expect and what we normally observed. When we had chronic long-term smoke effects, just all the time, Colorado, hefty background, but nothing, not hundreds of micrograms per meter cubed of aerosol, we would just see this sort of long, chronic, mild smoke. What we would see was a suppression of both photosynthesis and of monoterpene emissions. When we had a very intense smoke event, an acute event, extremely large amounts of smoke, very difficult to see across the road, sort of levels of smoke. What we then saw was that this would actually invert the temperature dependence of monoterpene emissions, and we haven't figured out the mechanism for it, but it tells us that the plants really, really do not they get very stressed with large amounts of smoke. So overall, this section, wildfire smoke exposure impacts plant health and I think potentially crop yields. Wildfire smoke also changes the natural VOC emissions and induces the uptake of some potentially toxic smoke compounds. Okay, the last part, don't worry, this is going to be quick, is about 90% of our time. And in this case, we can think about Cruella de Vil, one of my favorite villains, and many, many smoke VOCs that she might bring indoors. And the reason I think of this as 90% is that we spend, on average, Americans spend 90% of our time indoors based off of human activity pattern surveys. Some caveats in the footnotes. The challenge is that most atmospheric chemistry research has focused on outdoor air. And I think this is really important to put in context of how important indoor systems are. Rich Corsi, who's now at UC Davis, always use this context. And I think this is really useful. So if you think about the average life expectancy of an American, it's about 79 years. Based on activity patterns, we spend on average 69 years of our life indoors, 54 years at home, 26 of those years sleeping in bed, and about 4 years in vehicles. What this means is that most of our exposure then is happening indoors. But the chemical reactions that are driving indoor air are fundamentally different from those driving outdoor air. So this is what's driving our exposure, and we don't really understand what chemical systems control that exposure. So in order to think about this, my group has been working on a series of experiments, but the most recent one that I'm going to tell you about is CASA. And so CASA was the Chemical Assessments of Surfaces and Air study. And what we did was brought state-of-the-art instruments from over 13 institutions to the National Institute for Standards and Technology in Baltimore, Maryland, near Baltimore, Maryland, and in Gaithersburg. And what we did was use their house. They have this beautiful house on the NIST campus, the Net Zero Energy Residential Test Facility. And they use it to think about energy in buildings and how to measure energy. They agreed to let us come and do some chemistry experiments. So the goal of CASA was to really investigate how indoor and outdoor pollutants transport and transform across different phases, gases, particles, and surfaces in a house. This was a substantial group effort in 2022, and it involved a huge amount of measurements. We set up all of our mass specs in the garage of the house and pulled air in from the house into the garage. We also had surfaces, low-cost sensors, very high-cost sensors, bit of everything in the house. So the specific piece I'm going to tell you about is what happens when smoke gets inside. So the way we wanted to think about smoke, it turns out I have a student who spent a bit of time bartending during undergrad, and she suggested that there's this fancy device called a cocktail smoker. And what you do is you put in little wood chips and it heats them up to a good temperature to make a huge amount of smoke. We chemically analyzed the smoke and found that it was almost identical, both in terms of the VOCs and in terms of the particles, to the wildfire smoke that we were measuring during that weekend aircraft experiment that I first told you about. So we released the smoke. We did a series of different types of experiments. I'm just going to tell you about some of these direct releases. What we found is if we looked at the gases, we found that they were pretty interesting. This is work done by Jinan Lee, who actually got his PhD at Stony Brook and has been an absolute delight to work with here at CSU. And what Jinan noticed was that every time we added smoke, we would have an increase in different smoke VOCs, and then they would slowly decrease, but they would never return to background levels. And so what he noticed was that smoke VOCs persist indoors, even after days, and we calculated months and potentially even years after a smoke event. We would always see an elevated level. And what was happening is that these smoke gases are sticking to surfaces indoors. And Jinan was able to watch this partitioning for the air concentrations. So what we found is happening is that those smoke VOCs are coming into a house. And if we look at their signals in the mass spec as a function of time, what we see, if you just pay attention to that blue trace, is that this furan type compound would initially come into the house and it would decrease. And this is because it's getting adsorbed onto surfaces. We also could account for dilution and transport. So the surfaces were initially removing pollutants from the air. Second thing was partitioning. So then we would see the stage where we had uptake. There would also be some release as we move towards equilibrium, a constantly shifting equilibrium point. And then the third stage was one of desorption. And so we could characterize these different stages with different time scales that we could calculate relative to the ventilation rate of the house. And I'm not going to go into a huge amount of detail on these, but what I want to tell you is a couple things. First off, what Jinan was able to show is that chemical structure controls that partitioning behavior. So we were able to use these time scales at which the change in the VOC relative to the ventilation rate. We used an SF6 marker. At that exact point, that time scale was related to the vapor pressure of the compound, the saturation concentration. And so we could show that these timescales directly correlate for all sorts of different smoke VOCs to saturation concentration. What Jinan was then able to do was to use this relationship and predict what was going to happen and where all of these different VOCs were going to go. And so what he was able to do was partition out where these different VOCs went. And it turns out that most VOCs, as a function of octanol air partitioning coefficients, you can look at the more volatile stuff like ammonia on the left, that would still sit pretty heavily on the surface after a few hours. Some of it would be ventilated and moved outdoors, and then a large amount would also be in the gas phase. If you looked at stickier compounds like phthalates found in plasticizers, also heavily present indoors, you would see that those would mostly stick on surfaces and then slowly bleed off over time. So you have these high amounts of smoke, but the nice thing is we can give you at least a partial solution. So these smoke VOCs persist and they stay indoors. And if you have that empty house after you've added smoke to it a few times, then you have these high levels. And I should note, we added very small doses of smoke. The amounts, and we used data from various purple air sensors actually in indoor environments to figure out how much smoke we wanted to aim for. NIST was not excited about us contaminating their house. house. So we actually added very, very tiny amounts of smoke, far less than probably most of you had experienced this summer, unfortunately, due to those transported wildfires, just based on PM 2.5 concentrations that you observed. So these are small amounts, and we saw this persistent amount of smoke VOCs inside the house. We tried a variety of cleaning techniques. Long story short, air cleaners do have a very, very small effect, and that effect is only present while those air cleaners are turned on. In terms of VOCs, the moment you turn off an air cleaner, all of those VOCs that are stuck on the surfaces of your floors and of your walls just slowly partition back out. So air cleaners do not solve the problem. You can open your windows. That will ventilate and bring hopefully cleaner air from outside inside if the wildfire smoke event has passed. And that will drop all of those levels, but it only works for as long as you have your windows open. When you close your windows, again, all of those VOCs that are partitioned to the surfaces inside your house now just slowly bleed back out. The only way we found that had a permanent effect on the amount of those background smoke VOCs was actually surface cleaning. So dusting, vacuuming, and mopping actually removes that surface reservoir from the horizontal surfaces. You can't really mop your ceiling. And so what we were able to do was show that by removing that surface reservoir, all of those VOCs that we saw here that had adsorbed onto the surface in orange, so large amounts of those VOCs, what we then saw is that we could remove those. So here's what we found. Wildfire smoke forms reservoirs on indoor surfaces that can become persistent sources of VOCs. Cleaning surface can remove some of those reservoirs. So if we go back to this initial question that we posed of how do gases and particles from wildfire smoke interact with forests and buildings, I hope you realize now that smoke is an emerging villain of the atmosphere. It impacts human health, ecosystems, and climate. We can think about the Grinch who stole particles. We can think about how this villain interacts with leaves, and I think there are a lot of open questions there. And then I think we need to really start thinking about what's happening indoors. So with that, I want to thank the huge number of phenomenal people I have the opportunity to work with, as well as our funding agencies and the many science teams that help us put all of these data sets together. And I'd like to thank you for your time, and I will just leave some conclusions up in the next slide. Thanks.
All right, great. Thanks. Thanks, Delphine. I love your villains. They're awesome. Anyone have any questions? Let's see. I can see applauses. So if anyone has any questions, I don't know, you can either shout them out or raise your hand.
I have one. I put my hand up. This is Ernie Lewis. Delphine, wonderful talk. That was a lot of fun, and having played with dry deposition way back when, it was really interesting to see the new take. I like that a lot. We need to talk more. At a Gordon conference a couple years ago, when indoor air was becoming a very big topic, several people pointed out, and I'm not a chemist, so I'm asking for your comments on this, that cleaning was actually possibly a bad thing, in the sense of people would go in and take all these chlorinated compounds and mop their floor and do this and just form all sorts of nasties that didn't go away, and we're living inside breathing it. And they said, you know, just leaving your house dirty might be better for health effects, because yeah, it's a little dirty, but you're not making, you know, highly chlorinated and other nasty compounds. So I'd be interested in your opinions on that.
Yeah, and my group was one of the ones doing some of those bleach cleaning experiments. So absolutely. And so with those experiments, we found, yes, you add bleach and you do all sorts of strange, I mean, the indoor system is a multi-phase chemical reactor, right? So if you add something that's any form of oxidant, you have all of this material in all sorts of different phases ready to react. So you can generate all sorts of strange compounds. So indeed, I think at this point, it's very clear. You add something that's an oxidant in any form or highly reactive, you're going to generate secondary chemical products, and many of those are going to be toxic. So that's a bad thing. Also, something that was being noted by various groups at the time was that if you cleaned with any form of scented cleaner, yes, you were adding VOCs into the air. Those VOCs, though, I will say, are not necessarily have negative health effects, right? Pinene, limonene for pine scents or citrus scents. I think that's less clear, but you're certainly adding more VOCs into the environment. That said, what our work, I think, makes very clear is that some form of surface cleaning is necessary to remove air pollutants. So I think there's a balance. Yes, you shouldn't, unless absolutely necessary, use any form of oxidant or chlorinated cleaner. But surface cleaning, I will argue now, is definitely an important tool in our toolbox for improving indoor air quality. So I think really it's that the science is emerging. And so as we get more evidence and do more studies, we learn more. And so guidance is going to slowly change.
Great. Thank you.
Yeah, go ahead, Steve, and then Daniel.
Yeah, hello again, Delphine. Thanks for the talk. I'm wondering if you could tell us something about how you do the size resolved flux measurements. It seems like a tough thing to do.
Yeah, absolutely. And here, I'm just going to stop sharing my screen for a moment. So what we do for the data that I showed you, we use an ultra high sensitivity aerosol spectrometer. So this is an instrument that, well, Ernie Lewis knows more about it than I do, but we basically use scattering to figure out sizes of particles, and you can do that very quickly. So that's a 5 to 10 hertz measurement, and so that works for about 70 nanometers to about 1,000 nanometers. We get 99 size bins, which we have to average down because there just aren't enough particles in all of those size bins. And then we couple this to fast measurements of turbulence using a sonic anemometer. And then we use either eddy covariance or wavelet analysis to do an eddy flux analysis. So this is a fast flux measurement technique out of the micrometeorology world. It's really been explored in terms of CO2 exchange, but yeah, that's the technique that we use. The new approach we're using is to use portable optical particle sensors, which are little devices with much cheaper lasers. So they work only in a larger size range from a couple hundred nanometers up to a few microns. And so those instruments we've had to adapt pretty heavily. But they do give us those fast measurements that we need. And if we sit them next to a sonic anemometer, we can again do that eddy covariance. And I think it's becoming clear we need to do some wavelet analysis to catch those flux measurements. So that's what we're doing.
Okay, thanks.
Go ahead, Big D.
Hey, Delphine, great talk. Really enjoyed it. I have two questions maybe. I'm really intrigued about the partitioning story in the house. So the deposited organics and then how they come off with time and that they repartitioning, or you vent and then the vapor phase has a lack of them and then they repartition. Is it correct? So two questions. Is it correct to maybe state that the wildfires which impacted us, and we had, aerosols 130 micrograms per cubic meter here on campus, huge amount of gases. Actually, they all this wildfire smoke contaminated all houses on Long Island.
Yeah.
And if I look at the vapor pressures and the constant, I would maybe say now boldly, or I ask you, I'm still exposed to this smoke in my house currently, likely. So there's a long-term exposure for all, and many of this stuff is carcinogenic. And likely, since I cannot wipe my walls and ceilings, I'm still exposed to that, very likely. That was my first question. Second question is, yes, I clean the floor regularly, but of course you cannot clean the walls or ceiling. What is your take on using, ozone, brute force? I'm against these air purifiers, therefore I'm asking that. I advise against it because it's too high concentration. It's really not healthy. But if it's so bad, how about putting one in a room, leave the house, go in with a mask, vent afterwards, you hopefully broke all down to smaller molecules, they all evaporate, and then you get rid of this coating. So these are my two questions.
Great. Fantastic. Okay, so the first one is about whether or not you have a long-term exposure source now in your house. And unfortunately, our experiments suggest that yes, you do. I think one thing that's important to keep in mind is that surface cleaning will literally only take away the compounds that are adsorbed on the surface. And so one thing that Jinan was able to calculate was how deep some of these gases were actually penetrating. And we think that's really penetration into drywall or onto little reservoirs and pools on surfaces. And we only did these short-term experiments. We exposed the house for minutes, and then the smoke sat in the house for, again, maybe 10, 15 minutes. So these were tiny exposures relative to what actually happened. With that, I think so I guess my take on the chemistry is that if you can remove those reservoirs fast enough, then you can stop them from burying deep into your drywall. If you don't get that, then you have a longer source. Now that concentration might be lower, but you have a longer source. Now, the big question is that source enough to cause a health effect, right? And that we don't know. So we spent some time talking to folks at the EPA who understand a lot more about toxicology, and it's a very complicated problem because these molecules are weird, and they don't have the right animal model studies on all of them to know what the concentrations are. The larger issue is that we're also looking at a complex mixture of gases, and so in terms of health effects, it's not just your exposure to one nitrophenol, it's your exposure to all of these different VOCs at the same time. So we don't know. The toxicologists at EPA think we're a pretty long way away from knowing what is a dangerous amount. So I don't have any answer for you in terms of that, other than yes, there is a long-term source. On the flip side, Joost de Gouw's group at CU Boulder did some measurements after the Marshall Fire, and they think that those levels are not very high. The challenge with that, with an unplanned experiment, is you don't get a background measurement. So you don't know what the levels are before the wildfire event. So I guess we don't know how bad that is for you. I think your second question about ozone is a really important one. So ozone will certainly oxidize compounds. The problem is it will oxidize everything else in your house too. So all the plastics, all the other materials, everything else is going to also get oxidized. Those oxidation products are not necessarily all more volatile, right? Some of them can actually be a lot less volatile. And some of those oxidized, less volatile components may or may not be more toxic. So we know that ozone damages materials. We know that it will create some toxic products. So you're creating yourself a new set of compounds to be exposed to. So in general, I strongly advise against adding ozone. We actually did a lot of ozone additions to the house. That's sort of the next piece of analysis we're diving into, is to see what happens when you add ozone into the house. The first cut on the analysis is that the ozone has all these surfaces and all the VOCs that are inside a home normally. It's just this huge amount of reactivity. So we see all these effects, and we see a lot of reactions, and they're not necessarily all the ones we would have expected to have happen.
No, great, thanks. I thought so, but I wanted to hear a take. But so what I learned, if I design my next house, my whole room is tiled.
Yeah, absolutely. Just a
tile
big glass
everything,
house.
beautiful marble tiles, and then everything is okay. All right, thank you.
Great.
Your
Okay,
room is tight, don't breathe.
I got a question for you, Delphine. Why are the stomata closing in response to smoke?
Yeah, I think I don't know. My hypothesis is that it is a stress response. And so
what though do you think? Because
all of these toxics.
You think so? Because
well, it's either the toxics or it's the fact that you have so much smoke that there's not enough light, and so the plants just close up because there's no point. That said, there's a lot of CO2 that comes along with wildfire smoke. So what's interesting about this is people have modeled, based on some pretty sketchy background, but people keep on telling me, but smoke generates more indirect light and plants are more effective in the photosystem too. I think it is that uses the indirect light. And so they should actually enhance their productivity. But that's the opposite of what we're seeing. We see that those stomates close. So I don't know exactly what it is that's forcing the stomates to close. We spend a lot of time calling a lot of ecologists around the CSU campus asking what we had done wrong. And they all said anecdotally that that's exactly what they also observed, was that when there were smoke events, they were also seeing that the plant stomates would close and they couldn't do their experiments. Their response was, so we don't do experiments when there's smoke. My response to MJ was put on an N95 and see what happens. And so we don't know why yet. So I hope we can figure that out.
Yeah, because it's not a standard stress response, right?
Nope.
It's not water, it's not temperature. They're not losing water. So it's not clear.
But plants do some strange things in response to ozone. And so they don't necessarily stay open. It's, again, not clear. Literature is all over the place. But
ozone's tough though, because I think a lot of those studies, they were really hot days where they may have been stressed.
Yes.
But your plants might not necessarily have been. Maybe they were. I don't know.
It wasn't heat stress. It wasn't heat stress.
No, right.
The temperatures were actually quite reasonable. Yeah.
Right. Okay. Cool. So that's kind of interesting too. Any other questions? Okay, awesome.
I have one.
Yeah. Malcolm, go ahead.
Of those 69 years, the average person lives indoors. A lot of people in North America heat their houses in winter with wood-burning stoves. And from what you've been telling me in this fascinating talk, is that this could be a big problem regarding aerosols.
Yes, exactly. That is a very substantial problem, and it's a big concern. It's been known as a big issue in terms of people who use biomass burning as a fuel source for cooking. That's a sort of a well-established indoor air quality problem, but generally thought of in terms of particulate matter. Yes, what we're suggesting is that anytime those VOCs from combustion get into a house, they're sticking on those surfaces and providing a long-term, so not just while you're burning the wood. Yes, we think that that's potentially a long-term exposure source. Again, is that enough to have health effects? We don't know. But yes, that is definitely a problem that we're concerned about.
Thank you.
Yeah.
Even an elevated CO, long-term exposure to elevated CO to 400, 500 ppb, which is below, maybe your 1 hour threshold. But if you've got like a wood burning stove smoldering all night, that's generating parts per million to hundreds of parts per million CO. So if you can smell it, there's a lot of CO there, and that can give you headaches and stuff like that. I mean, CO is toxic.
Yep. And even high levels of CO2 cause sleepiness and decreased concentration. So definitely a problem.
Wow, so much interest in indoor air quality. I'm gonna try to sleep less. I don't want to spend 26 years asleep, but
really, I love sleeping. I think
I know, but you look at that number, it's like, oh gosh. Delphine, thank you so much. It was awesome. It was a great talk. I love your graphics, and thanks for joining us. I don't know. I'm gonna clap like this.
Great. Thank you so much. And really appreciate the questions. And if anybody has any more, always feel free to reach out, and I'm always happy to chat science.
I will be in touch soon, Delphine.